Okay, so hi, I'm Konstantin. I am a medical doctor and also a postdoc at the brain simulation section since 2020. And I just came back from a clinical rotation for 1.5 years for my neurological or neurology residency. And today I'm going to talk about modeling mechanisms of psychosis, or at least what we tried with our approach. And I'm sorry, this is now a bit out of place. We had to switch dates. So this would have fit perfectly after Leon Stefanovsky's and Leon Martin's talk. But I hope that you're still fresh into the semantic knowledge mapping theme, but I will also provide some background knowledge again. Okay, so just a quick reminder: the exam will take place on the 10th. It's on the from the 5th of December to the 10th of December. You will receive the exam by email, and then have 5 days to complete the exam. It's a multiple choice questions exam, and it's always just one question, only one answer is correct. And depending on your program enrollment, you get 2 or 3 ECTS points if you take part and if you pass, which I think if you give it some thought, you will probably manage. Okay. So as I said, the basis of what we did in this work is in more detail explained by Leon Martin's talk from week 4. And so I'm going to now focus on one particular use case we're using our framework for. And this use case is related to psychosis. So there's a question, one sec. No question, just a statement. Okay. And again, I'm a neurology resident, not a psychiatry resident, so forgive me if I get some of the DSM terminology wrong or something like this. But a point I wanted to make at the beginning is the differentiation between disease entities, symptoms, and syndromes. And I oftentimes, when I read especially neuroscientific papers, there's not much of a differentiation between these. So especially for these multifactorial, very complex diseases where the pathophysiology is just not well known, the whole disease will often be just described by one word, or there's no real grouping and no real differentiation what exactly we're talking about. So with this in mind, I will just do a bit of a detour. So screening for newborns. So what are we doing here? We are taking one sample of blood, dry it on this piece of paper, and then check it for genetic diseases. And why are we doing this? There's some diseases, some genetic diseases where the pathology or the pathogenesis is quite well known. So, for example, one thing we're checking here is a metabolic disease, which is called phenylketonuria. And in this case, one specific enzyme, the phenylalanine hydroxylase, or PAH, is not functioning properly due to a genetic mutation, usually by inheritance. So it's an autosomal recessive disorder. And if this happens, the essential amino acid phenylalanine cannot be properly transferred into the tyrosine, and more toxic agents of this phenylalanine will accumulate. And this leads, in the course of the disease, then to brain dysfunction, also to intellectual disabilities very early on. And if we know this, there's no special therapy, but if we know this, we can adhere to a special diet, restricting the phenylalanine uptake to serum level of less than 20 milligrams per deciliter, but still having more than 1 milligram per deciliter, because it's still an essential amino acid, and we have to consume some of it by our nutrition. But if we adhere to the special diet, we'll now have no adverse effects of this monogenetic disease. And another one, a bit more complex one, but which is also being tested since October of 21. So it's quite new in the newborn screening tests, is the spinal muscular atrophy. And in this disease, the SM1 gene is mutated. And this is the survival motor neuron 1 gene that encodes for the survival motor neuron 1. And this basically builds up most of the spinal motor neurons. So if this is not functioning properly, you will get atrophies, paresis, stuff like that also very early on. There's a second gene that's very similar, almost identical to the SM1, but it only produces around 10% of the functioning SMN protein, depending on the type of SMN2 gene you have. So you still have a rest function, depending on the type, even quite a normal one, but it's not as good as if you have the complete and functioning SMN1 gene. And this is also life reducing. So here in these black dotted lines, you see the expected life span without treatment. And in 2016, one of now many very specialized therapies has been introduced. This antisense oligonucleotide mucinazine, which packs itself into just after the exon 7, and then cuts out the mutated part of the SMN gene and restores most of its function. It's not completely the same, but still with a massive benefit on the clinical functioning of these patients. In comparison to the special diet for the phenylketonuria, it's quite expensive. So the therapy for 1 year costs about 300,000 euros, which is applicated in 5 different sessions. And I did this once, and it's really needs steady hands, because it's like a 5 milliliter ampule, and you have to apply it intrathecally, so inside the cerebrospinal fluid. Each drop you miss is around 1,000 euros, so you have to be very careful. Okay, why? Okay, so I'm not sure where I lost you. So yeah, so these monogenic diseases, the disease pathway kind of straightforward. You know, the gene alteration, you know, the downstream effects on the transcripted proteins, the pathways, and then you can also kind of explain how the clinical phenotype, so the symptoms, come to pass. And with multifactorial diseases like psychosis, for example, or Alzheimer's disease, it's not that simple, really. It's mostly a combination of very, that's what it's called, multifactorial, many different parts. So we have genetics, proteomics, epigenetics, also societal factors, and especially age and time plays a crucial role. And even the clinical phenotype is sometimes not as distinctive or as clear as we might think. And this is especially true for mental disorders, which are often lacking quantitative, all of them are actually lacking quantitative measurements, biomarkers that can differentiate between the syndromes or disease entities. That's why they are usually ordered into syndromes. And what is a syndrome? Syndrome is kind of a combination of different symptoms, right? So in this psychotic syndrome, it's a combination of these 5 psychotic symptoms. I will talk a bit more in detail later on. But what is the problem with this? As I said, the differentiation, not quality though, the differentiation with these syndromes or symptoms is not very specific. So if we see this graph here for all psychiatric symptoms, you see that around half of the graph is connected inside this one cluster. And if we look at the whole thing, it's more has a kind of small world characteristic with an average path length of 2.6 between two symptoms. We also see, if you can see it here, the schizophrenia syndrome is highly interconnected with, for example, mood disorders. And this we can also see in the different definitions or the different entities that can, different psychotic disorders that can lead to a psychosis, or that can be the reason for a psychosis. So we have schizophrenia, which is the most likely, the most probable, but then we also have schizoaffective disorders, bipolar disorders, even major depressive disorders, and that's just the psychiatric idiopathic primary psychosis, psychiatric disorders. We also have, if we see the clinical phenotype of a psychosis, it can also be induced by toxins, right? For example, the patient with Parkinson's disease who took too much of his dopaminergic medication, someone who took PSP, angel dust, can also lead to psychosis, sleep deprivation. And then the third part, which is just summarized here, psychosis due to medical conditions, which we also sometimes call organic psychosis. So in this case, and this is maybe for me as a neurologist a bit more relevant, there we have a disease entity that we can quantify usually, and that we can also treat therapeutically, that causes this psychotic syndrome or these psychotic symptoms. And why is it so important to differentiate between these different etiologies? I just wanted to quickly talk about the NMDA receptor encephalitis, which was discovered like 10 or 15 years ago, but is now a very highly researched field. In this case, the etiology or the pathophysiology is that you develop autoantibodies against the NMDA receptor. And this NMDA receptor gets internalized, which is important so it doesn't get destroyed, for example, as in other encephalitis, but it gets internalized and is not gone forever. So the trajectory, the remission rate of this disease is quite good, even if you have some cases where patients end up in the ICU. And it's often associated with a teratoma, so it's kind of paraneoplastic disease, we call it. Because the teratoma builds these autoantibodies, and it's also related to herpes viral encephalitis. And the cause of this disease is usually that it starts with infective, close to an infection or just right after infection, and typically causes a very fast progressing disease. So when normal psychosis in the course of maybe a schizophrenic disease or schizoaffective diseases takes time, about weeks to months before the psychiatric symptoms develop, and in this case it only takes a couple of weeks. In this phase, people with NMDA receptor encephalitis show these neuropsychiatric symptoms that can look like a, let's say, normal psychosis. And also the age when this disease happens usually is quite close to the psychosis. So it can be sometimes devastating that someone gets misdiagnosed with a psychosis on the basis of a schizophrenia and then actually has an NMDA receptor encephalitis. And usually it will always come out, so to say. So we will always discover this because in the course of the disease, the neurological complications start to grow. So the people will lose consciousness. They might have to be admitted to the ICU. And obviously the therapy is also completely different. So the psychosis of these patients doesn't respond well to dopaminergic medication or anti-dopaminergic medication, but rather you have to use immunologic medication, so something like steroids, intravenous immunoglobulins, or in the worst case, plasmapheresis, so kind of a washing out of the antibodies from the blood. And the very differentiating part from the psychosis used in the diagnostic of a psychosis induced by NMDA receptor encephalitis to the psychosis that is caused by schizophrenia is that we have this kind of quantitative, in this case also pathognomonic laboratory antibody that we can test. So this GLU-N1, which is a subpart of the NMDA receptor subunit. Yeah, just to show that it's not just the NMDA receptor encephalitis that can cause schizophreniform psychosis, but a lot of different encephalitis. But this is rather rare case. It's just to show that the reason why someone can show the clinical phenotype of psychosis can be very different, can be very multiform. Okay, but now to the psychosis in the most common case, so the so-called idiopathic psychosis, which basically just says we can't find another reason for it, so to say. And the typical symptoms are these highlighted symptoms here. So delusions, something like people thinking they're being persecuted, or delusions of grandeur, hallucinations, typically auditory hallucinations, which show in around 3/4 to almost 100% of the patients with psychosis in schizophrenia. Disorganized thinking, so formal thought disorders, kind of rapidly switching between topics, derailment. The same for motor behavior, so which can lead almost to catatonic motor behavior, so not moving at all, kind of staying still with your movement. And then so these are positive symptoms, and then the bit more difficult aspect of the hallmark symptoms of psychosis, the negative symptoms, which sometimes seem or can seem like a affective disorder, like a depressive episode or depressive disorder. This is also the problem, because this has such a high overlap with affective disorders or mood disorders, that people with primarily negative symptoms usually get diagnosed way later than people with positive symptoms. Yeah, and if you want to diagnose, as I said, this is from the DSM. I'm not a psychiatrist, but if you want to diagnose someone with schizophrenia, he has to have at least one of these 3 positive symptoms, and also the whole thing for at least a period of 1 month, or a significant portion of in the period of 1 month. Then it has to have a qualitative effect on his social performance or his social life, on his functioning. He has to have some sort of attenuated disturbance for at least 6 months, and then inside of these 6 months, at least 1 month of full-on positive symptoms, so one of these 5 symptoms, or at least criterion A, so one of the first 3, or one of the first 3 and 4 and 5, or all 5 of them. And we have to be sure that it's not, for example, a psychosis due to other organic reasons or other medical reasons, or toxic psychosis. Or in this case, or if you're on the psychiatric disease entities, also not caused by depressive or bipolar disorder. And the cause of the disease can be very different. So you can have kind of a continuous schizophrenic disease. You can have recessive or episodic course, and so on, and everything in between. This. And the main point here is that we don't have any quantitative measurements. We have to make the diagnosis of schizophrenia clinically. And when we talk about the epidemiology of schizophrenia, so the lifetime prevalence is around 1% worldwide, which is quite high. And there's no real difference between men and women. And the age of onset is quite young, so something between 20 and 30. And it's associated with a lot of annual costs. If you take the USA, it's around 150 billion, which is partly caused by the treatment costs and the hospitalization of the patients, but mostly by the inability of these schizophrenic patients to work in the long term, usually. And the onset and progression of the disease is usually partitioned in these first wave, second wave incidents or hits. So you don't just, so you need to kind of have this first genetic or epigenetic hit, right? With infection during childhood, hypoxia during childbirth, your genetic load that you just inherited. And then, but this alone doesn't assure that you get this psychosis later on, or that you develop schizophrenia in the later turn of the later course of your life. You need to have this second hit, which are mostly environmental factors, and then epigenetically can affect your proteins, your transcription of genes, and so on. And some of these are drug abuse, social isolation, but also socio-demographic factors like migration, or where, or if you live in a big city or not. And if you both receive these first and the second wave hits, then your first psychotic episode can start. And usually it has this, can have, as we saw, can have different time courses, but usually doesn't end with only just one episode. Only, what do we say, 50%? 25%, yeah. The other 25% have a chronic disease, and 50% have more than one episode. Okay, and if we then look at this graph from a different point of view, so kind of in the point of view of fundamental brain development, then we have this first part in the first hit is happening in the brain formation. So this part is also more strongly influenced by your genetic load. And the second part, the brain reorganization, that is why also environmental factors, drug abuse plays such a big role in this second hit phase. And then this green epilepsoid is the so-called window of opportunity. So the therapeutic window where everyone wants to find out who's someone who might develop a psychotic disorder, psychosis, and try to intervene before it actually comes to pass. So to determine who is at very high risk to develop a psychosis, and then even just intervening in his drug abuse, or trying to relieve some of the social stress to stop this first psychotic episode to ever occur. Because once it occurs, it's kind of hard to get rid of it, right? And yeah, this is also, so right now the therapies usually focus on the disease when it has already taken place, or when it's already active, right? And these are then symptomatic treatments. So because you know that a overload of dopamine is causing most of these positive symptoms, antidopaminergic medications usually administered here. And what's called here the tomorrow's therapies are looking at this window of opportunity and looking at therapeutic interventions that can actually inhibit the first episode of psychotic symptoms. And then, of course, if we would have some sort of specific schizophrenic or psychotic genotype, we could then start even earlier. But we will see in the course of this talk that it's not that simple, really, to define a genotype for schizophrenia or for psychosis. Yeah, and this window of opportunity, we are trying to reach these individuals or these patients, not yet patients, at clinical high risk for psychosis. So people that are very likely to develop a psychosis in the course of the next weeks, months, years. And the points that are pointing into that direction are again clinical phenotypes, not quite yet the complete picture of a psychosis, but something a little bit before that. So kind of attenuated psychotic symptoms, or brief intermittent psychotic symptoms, but not in a length that it would constitute a full psychotic disorder. Or if we know that there's a high genetic load, for example, because both parents have schizophrenia, or we did some genetic testing for whatever reason, and we know that the likelihood of someone developing psychosis is quite high in this sense. Yeah, just to see that, you know, these attenuated clinical high-risk symptoms usually come before psychosis and also in a different time manner. And here's again the window of opportunity we want to intervene. Yeah, we talked about already. Okay, so we have this window of opportunity, which is important. We have this clinical high-risk individual group, which is important in the sense that we have to, that this is the group of individuals that we will most likely have the biggest impact on with further therapies or future therapies. And but to understand why or how we can intervene in this phase, in this window of opportunity, we of course have to understand a bit more of the pathophysiological features of psychosis or psychosis in a or schizophrenic psychosis, for example. And there's a lot of different theories going around, but it's kind of condensing on a few theories that we get more and more evidence for. And one, so this is from 2018. It's a bit more refined now, but overall this is a quite nice overview. And it starts in this depiction of the pathophysiology. It starts in the hippocampus with a alteration in NMDA receptor activity and also in the GABAergic transmission. So we have, for some reason, this can, I can explain later what these reasons can be. We have a malfunctioning NMDA receptor and also malfunctioning GABA receptor or less GABA production or in the synaptic cleft. This then leads to a hyper excitation due to glutamatergic pyramidal cells, which then, at least in this theory, lead to a higher dopaminergic production of striatal dopaminergic cells, and also in the midbrain, and then also leading to higher dopaminergic production or activity in the prefrontal cortex, which usually leads to these positive symptoms. And why does this theory of like interplay between dopamine, glutamate, and also GABA maybe make sense? First of all, if we have, with the psychosis syndrome already started, we know that dopamine antagonists work. So the role of dopamine, at least in the active phase of the disease, is a kind of given. And then also with glutamate, as we just learned the anti-NMDA receptor encephalitis, where we just have this very clear pathophysiology, also leads to psychosis in one part of the disease, in the course of the disease. Yeah, but we don't have that kind of perfect allocation, but it still makes sense if we think about the interplay between interneurons and pyramidal cells that GABA also plays a role. So overall, we have an altered excitation inhibition balance in different parts of the brain caused by different neurotransmitters. One point about the genetic factors in psychosis and schizophrenia, it's, you know, that it's a quite high, well, genetic factors play a high role because of the concordance between identical twins. So 50% concordance between identical twins means that if one twin develops psychosis in his life, it's a coin flip if the other twin also develops psychosis. And also the parents with psychosis usually the rate of the children developing a psychosis is much higher than for normal individuals. And there are two kind of thoughts about why this is the case. So one is this common disease, common allele hypothesis, meaning that there's not really one gene or just a few genes who are using this, who are playing a factor here, but it's actually like a larger set of genes that all have a very limited penetrance in the course of the disease. And the other school of thought is a common disease rare allele hypothesis. So you need a very specific mutation in one of these genes with a high penetrance to actually have any genetic effect on the disease. Is there a question? Let's see if we have. Right, and there have been large genomic studies, GWAS, and basically the result really supports the common disease, common allele hypothesis. So there are a set of genes that might play a role, or that do play a role, but it's a large number of genes. So it's around 100 risk genes, and all of them play a small part in the pathogenesis of the disease, but we can't really pinpoint it to, like in a monogenetic disease, to the schizophrenia gene, for example. And some of these genes are related to glutamatergic signaling, so we kind of get the hypothesis gets backed up by GWAS studies and back and forth. And yeah, we talked about this already. And other protein and gene combinations that get highlighted by these GWAS studies are related to neuroimmunological proteins and genes. We'll talk about that later. Yeah, talk about this. And gene alterations aren't the only thing that we can see in the course of the disease. There are also brain morphological changes, especially later on in the disease when schizophrenia is completely there. And usually most of these morphological changes show some sort of atrophy. So for example, here temporal atrophy, enlargement of the ventricular system, which is always a sign that the gray matter has reduced in size, and so the ventricular system has more space to or can take up more space, let's say like this. And also new studies with specific PET imaging show that, for example, the synaptic vesicle protein 2A density is decreased, especially in the frontal, prefrontal areas in patients with schizophrenia. Yeah. So it's again another part of the puzzle. Oh, that's nice. And as I said, from these GWAS studies and also by animal studies and so on, we also know that inflammation plays an important role. Maybe also kind of initializing role, as I said, with the disturbance of NMDA receptors, for example, in the hippocampus. Maybe this is caused by some sort of chronic inflammation. Here, one work I actually found quite convincing, because it also takes into account the temporal development of this disease, is by Haroun et al. And they hypothesized that inflammation, which isn't always a bad thing, obviously, but chronic inflammation leads to inflammatory cytokines and tumor necrosis factor activation of microglia and macrophages, and the cause then to activation of these ROS, so reactive oxygen or nitrogen RNS species, and they, if they act for a prolonged time, can be neurotoxic. And overall, this leads to a synaptic overload with glutamate, and synaptic glutamate spilling, then also activating extrasynaptic NMDA receptors. And overall, to a case of kind of this excitotoxicity, partly caused by reactive oxygen species, but also partly caused by this overproduction of glutamate. And then in the course of this disease, or in the course of this chronic inflammation, so this is the early stage, this leads to atrophy or dying of astrocytes. This leads to downregulation of the glutamatergic signaling. And this glutamatergic signaling is obviously greatly connected to also GABAergic interneurons, or basically to all excitatory interaction to other neurons, and less glutamatergic pyramidal neurons will also downstream lead to more dopamine production, in this case, or more dopaminergic kind of disinhibition of dopaminergic neurons. Okay, so a lot of different puzzle pieces for the pathogenesis of schizophrenia. Firstly, it's still diagnosed clinically, so there are no biomarkers yet that can be used. There are a lot of different genetic or environmental risk factors that play a role in the disease trajectory. And there's a multitude of different hypotheses and proposed mechanisms that play a role in different parts or in different times of the disease and in the course of the disease. And part of these are neurotransmitter disbalances, and also neuroinflammation is one key pathogenic mechanism. Okay, so we're quite far along in the time, so I'm gonna speed through this part. So because it is such a multifactorial and so hard to understand and hard to pinpoint disease, there are obviously efforts to use computational modeling approaches to understand psychosis of schizophrenia better. And these follow different paths, right? So one is the top-down approach, the deductive approach, where you have a specific theory about the pathogenesis of the disease, and then build a hypothesis, try to conform it in your models, usually algorithmically. And then there is the oftentimes like data-driven bottom-up approach, that you have a lot of data, you have a lot of observations, try to define a pattern with, for example, machine learning, and then build your theory on top of that, right? And this is also important to these computational models because, and this is just kind of a reiteration of what I already said, the specificity of the clinical diagnoses of mental disorders is not very high. So you see here the kappa is a statistic for inter-rater reliability. And for example, if you have a disease that has like a rate of 10% in the general population, and you have two different raters that diagnose correctly with a rate of 85%, you get this kappa of 0.46. So not bad. So it's still a good agreement, but if you look here at major neurocognitive disorders, the correct identification of the this disorder is way higher. And we need to support this with biomarkers, with endophenotypes, with better models. Yeah, so bottom up, usually data-driven, top down. And then there's this third part that can help and partly skip this, maybe also this hypothesis generation, so knowledge-driven models that try to use the knowledge that's already there to help in the understanding of the disease. And this is also what we are trying to do. So we're trying a knowledge-driven approach. And as I said, we need these kind of, we would like to have different biomarkers especially for this vulnerable phase, for the window of opportunity, for the transition to psychosis, or then the transition to schizophrenia, that we can catch these clinical high-risk patients at the right moment and then actually intervene before it's too late. And there's multitude of different neurophysiological, but also brain morphological, genetic, blood values that are proposed, but none has actually made it into clinical practice so far. Yeah. Same thing. So we want biomarkers or even endophenotypes, which are kind of a even stronger biomarker. So it's just not just a like a quantitative lab value, like a serum biomarker in the blood, but it's actually something that is defined as a kind of own disease entity almost. So it can segregate between illness in the disease in the population. It can it's heritable, and it's usually not state dependent. So an endophenotype shouldn't change in the course of the disease. Right? So it shouldn't endophenotype should be something that's stable, even if we have the clinical onset or before or after. Something that we can quantitatively pinpoint the disease to, and obviously the benefit of this would be that we cannot just differentiate the disease in between, like, patient has symptoms, he's affected and unaffected, but have actually a trajectory and can way earlier define who's at risk and who's not. Okay. Yeah. So this is a study that we our collaborators, so study which we use the data from to build a use case for TB base. And in this study, they used a multimodal machine learning approach with different models, and also try to scale it clinically to try to predict in a set of individuals with clinical high risk of developing psychosis in the next couple of months, years, to predict who is transitioning to psychosis and who is not. And this was a European large multi-center study with 668 subjects, half of them these clinical high-risk patients. Or rather, so half of them were clinical high risk, and the other one was recent onset depression, again showing that the distinction between who's at risk of psychosis is not that easy, and the reasons why someone is developing psychosis can be very multifactorial. And the models they used were firstly clinical neurocognitive tests that were done every 3 months, basically. Then initially, and after 9 months, also an SMRI with volumetric imaging and a polygenetic risk score. And this was completely data-driven, and as I said, they try to firstly predict who is transitioning to psychosis, or to find predictors of developing psychosis, and also, as I said, as biomarkers haven't found their way into clinical practice yet, try to find a, what they say, clinically scalable optimal workflow. So something that can be used by clinicians in a time and also cost-sensitive manner. And in this case, the transition wasn't defined as having a full-blown psychosis, but actually reaching at least one positive symptom in this specialized psychosis rating test for at least 7 days. Okay, and what we see, so they followed this up for 3 years, and in this cohort of 334 patients, 26 developed psychosis or transitioned to psychosis in their definition. So not that many at all. And what we also see is the higher amount of comorbidity, right? So a lot of the patients also have a major depressive disorder, obviously because also recent onset depression was part of the patient group, but a lot of also affective disorder. Only few really substance use disorder, which was surprising, but yeah. And of these 26 patients that transitioned in this period to psychosis, 23 were of the group of the clinical high risk patients, and 3 of the recent onset depression. And in follow-up, 8 of these 26 patients actually went on to develop a schizophrenia. And this is the machine learning workflow. And basically, this was done in multiple steps. So first of all, at each step, they used a leaf site out cross-validation. So they had this multi-center study with different sites all over Europe, and then always left one of these sites out and tested against it to not get any epidemiological or region-based influences on their models. Then they did models for each of their 3 qualities. So the clinical neurocognitive risk calculator model, the polygenetic risk score calculator, the SMRI risk calculator, and then also built a stacked risk calculator where they stacked all these 3 models together in different ways, and then also a sequential risk calculator where they try to, in the sense of actually using this in clinical practice, try to only add one more modality if it's really necessary. So if after the clinical neurocognitive risk calculator, it's clear that he will most likely transition to psychosis in this period, we don't have to use the PRS or the SMRI risk calculator. So this is the first step, finding this optimal sequential stacked risk calculator. And then in the second step, checking for co-founders, trying to make this as lean as possible, and also then comparing the model to a complete new data set that they didn't include in the test or training set. And 2 feature metrics that they used was once the cross-validation ratio, which basically said that in this, for example, for this SMRI data, which voxel was statistically significant to predict the transition to psychosis in this whole cohort. And this was going 2 ways, so it could be negative and positive, and the sign-based consistency was basically the same, but only testing if this was statistically significant. And what they found for the clinical neurocognitive model is that they could reduce the initially 141 features to only 7. And if we look at these, so this is from these psychotic tests batteries. You see most of them are referring to positive symptoms. So the grandiosity, so delusions, perceptual abnormalities, hallucinations, So these were the most positive symptoms were the most differential when looking at the clinical model, and were also the most differential when looking at the difference between clinical high risk and recent onset depression patients. The time, okay. And the polyclinic risk score model also worked on its own, had an accuracy of around 65%. And the SMRI model also worked quite well. And what was interesting here is that look at the cross-validation ratio, it's a bit counterintuitive. So blue areas are actually volume increases and red values are volume decreases. And while it's no surprise that people that transition to psychosis have areas of volume decreases, right? So atrophy, what I showed earlier in this morphological brains changes in the course of schizophrenia. We also see a lot of areas, or just as much areas, that show a volume gain or volumetric increase. This patient, and it's also the shows the same importance for the prediction of for the onset of psychosis. And when we stack all these things together in this order, so ratas was actually another variable where they just asked the clinical expert what he thinks is going to happen, if he's going to transition or not. It's kind of a coin flip. Then the accuracy of the overall model was 82.9 or 85.5 with the ratas involved, which they then called cybernetic model. And we saw that most of the transition individuals or patients were actually part of the clinical high-risk group, but curiously, if they removed the recent onset depression patients, the performance of the whole model was near zero. So they lost the whole accuracy of the model. So they somehow the group of recent onset depression patients has a very important part in this pathophysiological understanding of the disease. Yeah, here again, the accuracies of the single models, also of the stack model or of the optimal sequential model. And not surprisingly, the multimodal models outperform the single models. Okay, but which is surprising is that the sequential model, where only in around a quarter of the patients all 3 or 4 modalities had to be used, also outperformed the stack model, which was always using all modalities. Okay, so I said we use this as a basis for use case for TB-Base, but what exactly do we do here, and what can we learn from this study? So we found these features that are predictive of the transition to psychosis, but especially the SMRI features. We do not know what's happening there, what's causing these morphological brain changes in this short period of time. How can we define mechanisms behind these volumetric brain changes? Yeah, and this is the use case we did. Because it's such a long time ago, we're just gonna recap shortly what TB-Base does. Basically, TB-Base is a knowledge mapping tool where we use the literature mining knowledge base SkyView, which has the whole PubMed literature database in a literature mined annotated fashion, which means that each term that can be referred to one of the mostly used biomedical dictionaries or ontologies is annotated in this literature corpus. So you can use free text, but you can also look for specific entities from specified ontologies like the MASH ontology, the medical subject headings. And what's more, you can also, each anatomical term is annotated in the Uberon ontology, which is an anatomical ontology. And what this lets you do is that you can search for specific abstracts that mention specific annotated concepts in the literature. And what we then did, because there's no connection between this semantic anatomical ontology, or there was no connection until now, we built a transformation matrix for each anatomical term in this probably most biggest anatomical ontology out there, which cross-species references, and so a lot of hierarchical information, and then use these anatomical semantic terms and map them in a very tedious and long manual creation process onto a brain atlas. And there we use the Glasser parcellation because it's one of the most refined and also functionally distinct brain parcellations. Then mapped each of the Uberon terms that we found in the literature to brain parcels. And this then allowed us to when we query any concept in the sky view, to give us a list of associated Uberon terms. Oops, rated by their relevance, and for this relevance measure, we use Skyview uses the Colbert-Leiber divergence, which is a distance matrix, which basically tells you how relevant is that brain area in this query that you use. For example, when you're looking for glucose metabolism in Alzheimer's disease, how relevant is this brain area compared to all the mentionings in the whole literature, right? And we compare these then relevancy maps, so semantic relevancy maps, to different empirical maps from neuromaps, which was a large like where they combine different modalities from very different fields in a common brain parcellation atlas. And we saw here that we can have a good correlation between MEG rhythms, but also for PET tracers and also for metabolic features. So very broad spectrum of possible uses of TV-based maps, and most of them seem to display empirical features in the way in the cases where we can actually test it. Okay, so we have these TV-based brain maps where we can query a specific context, specific concept, and which gives us as an output a brain map in the Glasser parcellation, telling us which areas are important to that query. And now we have this SMRI-based model from the study I just showed you from Potsolaris et al., which shows us a differential map of volumetric increases and decreases. And what we then set out to do is try to use these TV-based maps, especially brain maps of gene or protein entities, to decode this pattern we saw from Potsolaris et al. And in order to kind of limit our feature selection, we used a knowledge graph that was built by Fraunhofer Institute again, which by literature mining built a knowledge graph or built triplets from the literature with including all concepts that are related somehow to psychosis, basically. And what they did, they used literature mining to go through all abstracts, through all PMC articles, and sort out these triplets formed of associations to pathways or to genes to biological processes that are implicated in psychosis or in psychosis inducing diseases like schizophrenia or bipolar disorder, and then use their own biological expression language to build such a knowledge graph. So, for example, here in this abstract, we see that NMDA receptor decrease in the PFC and energy decreases green 2C. Yeah, okay. Yeah, green 2C. Sorry, I had to think about that a second. The green 2C is a subunit of the NMDA receptor. We see here from this abstract that there's a relation between the NRG1 protein and the subunit of NMDA receptor that is defined by a decrease. And with this triplets, Fraunhofer then built this whole knowledge graph that kind of just combined all of the semantic information, the literature out there about anything that's related to psychosis, basically. And this didn't just include genes and proteins, but also biological processes, mostly annotated in Go, also biomedical terms annotated in MeSH, and chemical compounds and complete disease entities and everything. So kind of very widespread widespread knowledge sources. And we kind of limited ourselves to, as a feature selection mechanism, to only proteins from these different features, or proteins or their encoding genes. Because we thought about how can we understand this structural spatial structure from the Consulara study. How can we explain it in a way that might lead to an endophenotype, or that might lead to a biomarker that we can actually understand? And we include again too many knowledge sources of too many different layers that are not very easy to understand their connections. We might lose explainability. So we limited ourselves to genes and proteins to find this kind of intermediate endophenotype to explain this SMRI phenotype, right? Yeah. And just to reiterate, so if we are limiting ourselves to the knowledge source of genes and proteins, this is still not an easy feature selection we have, right? So we have the whole genome, and we have of that genome all genes and proteins that are actually involved in known pathways, and these are collected in pathway databases. And now PsychoMM6, so this knowledge graph, uses part of these pathway databases that are related to psychosis, right? And the thing is, so part of these pathways are already outside of pathway databases, and we have no knowledge about the true psychosis pathway and how much of this we are already, which is already inside of a known knowledge about pathways and disease-specific pathways. Yeah. So this is kind of what we know. This is what we know we don't know, and this whole part here is what we don't know we don't know. And if we then look again at the information that we extract from this knowledge graph, so the spatial distribution of these genes or proteins on a 3D brain, we then again have only part of this pathway knowledge that we have. We have only the knowledge that is already associated to brain anatomy in some way, right? So again, we're very aware that we are dealing here with limited knowledge, and also maybe just things we don't know. Okay. Yeah. So the first thing we did, so we thought about, so we used the knowledge graph as a feature selection mechanism. extracted all the genes and proteins that were relevant for psychosis, then mapped them, and then realized, okay, we have this relevancy metric, which is not very comparable between different brain maps necessarily. And so we thought, and which is also heavily influenced by publication bias, right? So the hippocampus is just an area that is highly used in scientific literature. So we said that to kind of battle this also publication bias and this bias by the relevancy, we just binarize brain maps, the protein brain maps, saying that an area that has a relevancy of more than 1 or more than 0 is relevant, and if it's 0 or less, it's not associated with the disease and the not relevant for this for our task. Okay, then we used, with the help of Kirat especially, we used a linear spot vector regression, and this we chose because it's quite easy to understand what the weights of the regression mean, right? So a high weight means high importance to the decoding problem, and we can also have positive and negative weights. So this gives us more freedom of rebuilding this transition map. And what we see here briefly is that even if we use the whole genome, the reconstruction is only mildly better than if we limit ourselves to the psychoanalytic, and if we look at a effect strength like the class delta, which is the mean of the experimental and control group divided by the standard deviation of the control group, you see that the class delta actually doesn't improve when we use the whole genome. Right? And then as a next fine-tuning mechanism, we then used a cross-validation method in the same way that they used for their study. So we used to leave one side out cross-validation. And what we saw here, that the predictive capability of the model dropped slightly, but actually did not significantly change the order and set of genes. Yeah, we also optimized the feature or the map that we were actually trying to decode by using the sign-based consistency as a mask for the CV ratio, just to be sure that we only look at voxels that are super important. And here's the original CV ratio in a past-related form. And this is our predictive, or our map that we rebuilt with our GNAPs. And another thing we thought about, so, and what also Kira talked about in his talk, if we have these problems, it's not always about achieving the best fit or the highest R squared. So we tried also different set of features which were better at rebuilding the map, but had no explainability, right? So sometimes we just want to use this machine learning tool or this machine learning method to find a relevant set or the most relevant set for our problem. So explainability is the main goal here, not the actual capability of rebuilding any spatial map. And with this in mind, we used the minimum redundancy, maximum relevancy idea to limit our set to so few features as possible, or as we need to rebuild the transition map without losing performance. And another thing we noticed, it's now going very into detail, but when we binarized the maps, obviously we lost some bit of information, and a lot of maps then kind of showed the same spatial pattern, because we discretized it only to 1 and 0, and this led to feature twins, so to say. And sometimes these feature twins made sense that they were also were involved in the same pathways or in the same biological processes, but sometimes they were just unrelated and just had the same spatial pattern in the literature. But overall, we saw that for the SVM, there was no distinction. We couldn't say which feature twin is going to this, and we couldn't say a priori which feature twin is more explanatory for problems. So we just kind of submerged them all into one feature, gave each one then partial feature importance. Another thing we noticed is that when we iteratively, so we use the forward feature selection method, so iteratively always adding one more feature and then doing this for 100,000 times, we saw that the model performance kind of peaked after including 40 features. So we only need 40 features to have the highest performance of the decoding model. And that's why we also limited our explanatory efforts then to the top 40 features. And if we look at these, so this is the selection frequency. So how many times this feature map was chosen in the n runs we did. And we see the decreasing importance of that feature. And we also see these feature twins that kind of identical twins that make up this singular feature. And now this is only 40 features comprising about 76 genes, but it's still really hard to grasp what's actually happening here. So we needed even more knowledge. We needed even more help to understand this, the mechanisms that are actually happening here and that are comprised in these gene maps. And for this, we used pathway enrichment. And this is basically, again, using vast knowledge and vast databases, for example, here. So these are our features, size corresponding to the feature selection frequency. And these features, for example, you can try to enrich with these proteome maps, which are but are basically only based on one pathway database, the KEGG database, and also where the protein names come from, the PAX database. But we also see some. So we see that we can actually group these into different biological processes. But to get even more information, to get even more knowledge, we use the string database, which is basically the most biggest database on protein-protein interactions that there is. They also use KEGG, but they also use wiki pathways, reactome, so every information that is out there for human protein-protein interactions. And what you do here, you input a set of proteins. So in our case, these 76 proteins, and string then tells you if the number of edges of the kind of interaction you get inside of your data set is more than what you would expect from a random gene or protein set of the same size, right? This is what this enrichment value tells you. So, and in our case, the enrichment value was highly significant. So the chance that we get such a highly connected protein-protein interaction network was far more than chance. And what you can also do then, so you have then one network with all of your protein huddled together, but you can also then use clustering algorithms to further disentangle these networks. And what we use here is the Markov chain or Markov clustering algorithm, which was shown to be the best for disentangling protein-protein interaction networks. And basically, you have this inflation parameter that is based on random walks, but using a flow instead. And the higher your inflation parameter, the more strong connections get emphasized. So the higher your inflation parameter, the smaller and the more huddled together your clusters will be. And we then did this for our data set. So these are the 4G features which come to 75 genes or proteins. And for different inflation parameters, we then saw which kind of clusters built and what kind of association these clusters had to biological functions or to pathways. And with a 1.4 inflation parameters, the cluster with the highest significance, in the sense that it had the highest overall selection frequency under these within these features, was associated to interleukins, inflammatory response, and also to neural growth. And if you think back like an hour ago, we also talked about the mechanisms that are discussed in the etiology of psychosis, and inflammation was definitely one of those. And the second highest cluster with this inflation parameter was dopaminergic and glutamatergic signaling. So down here you see the glutamatergic signaling with AMPA and NMDA receptor subunits, and up here you see dopaminergic signaling proteins that are still connected. And the third and most important cluster with this inflation parameter was GABAergic signaling. So all of these mechanisms are not new, but it's quite interesting that we find them when we are decoding this completely new empirical data, right? And we did this for different inflation parameters. I'm going to skip over this, show you this Sankey diagram. So within this range of inflation parameters, we get meaningful clusters. Before that, we just have one cluster, and after that, the clusters kind of dissipate, and there's no functional or significant enrichment anymore. And what we see here, we see this big neural growth cluster that then dissipates into notch signaling, which is also a signaling pathway associated with neurogenesis, and also what do they call it? I think it was later on. And also dissipated in this neural growth and immune system pathways. And if we think back again, what are we actually modeling here? We are actually modeling volumetric brain changes in an SMRI with volume increases and volume decreases. So it makes sense that we have pathways, that we have explanatory mechanisms that could actually lead to such volumetric increases and decreases, right? So neural growth totally makes sense in this way. And of course, we also see, or not of course, but it's nice that we see these neurotransmitter pathways that are widely discussed. So GABAergic, glutamatergic signaling, dopaminergic signaling, and also some cholinergic signaling. Plasticity, that's the thing. That's not also associated with this plasticity. Okay, so I'm not sure if you could follow all of this, but if you forgot what mechanisms we discussed before, we went into this decoding problem completely agnostic of any prior knowledge, right? We had this empirical structural MRI pattern with volumetric increases and decreases, and then used TV-based maps of genes and proteins with the idea that if a gene or protein is relevant in a brain area, it also has some sort of function there to decode this mechanism, but still ended up with the same mechanisms that are discussed in the pathogenesis of psychosis, which is very nice. So neural growth we actually didn't so much talk about, but also makes sense in the sense of volumetric gains. Inflammation we saw, and this neurotransmitter imbalance, right? And obviously this has therapeutic implications. So for the inflammatory pathways or the inflammatory mechanisms, we can think about inhibiting these neuroimmunological processes. So, for example, interleukin-6 played a pivotal role, like in a hub of this inflammation pathway, which also has an antibody that already is in use with the inhibition of this particular interleukin. But obviously, with the caveat that we know that we have to know when to use it. Compt, yeah, just to show that the dopaminergic pathway and the antidopaminergic medication obviously still makes sense. And then BDNF, which is also closely related to plasticity and neurogenesis and neural growth, that could be a target for stimulation treatments. That's just the mouse cortex where we see that inflammation goes down when we apply tocilizumab. Completely unrelated, actually. Yeah, now I just want to talk briefly about something that we've been working on like right now. So it's some of these results are really from today. So if there are some logical mistakes, please excuse me. But one big limitation of our TV-based maps is that we do get these spatial patterns of relevancy, but we are not sure in every case what this relevancy means, right? So does high relevancy, for example, in this case, right, we are querying Alzheimer's disease and glucose metabolism. Does high relevancy mean increased or decreased glucose metabolism? So from our understanding from the disease, we would assume that it means decreased glucose metabolism, but we can't be sure. So as an extension, we used large language models, in this case ChatGPT, to interpret the exact semantic association that we are extracting here from the literature. So we went for this map through all, or ChatGPT went through all abstracts and saw how the exact connection between the brain area and the process, in this case glucose metabolism in Alzheimer's disease, is related. And in this case, these two brain areas are mentioned, temporal lobe and angular gyrus. And through some corners, we can extract that the glucose metabolism in these areas is actually decreased in AD. And if we sum up all this information from all abstracts that constitute this map, we see that it's actually how we thought the glucose metabolism in AD is really decreased in this areas of the map that are relevant. Yeah, you see some differences here in the maps. This is because we use the prompt in the way that if it's not mentioned, if there's some directionality between brain area and the context question, we just kind of rule them out. So we kind of ignore them for this directional map. They're still relevant for the relevancy map, but not for the directional map. And the same thing we can obviously do for all of our maps, and in particular also these gene or protein maps in the context of psychosis. Right? So this is the overall comp map. This is the comp, so the catechol O-methyltransferase protein enzyme in the context of psychosis. And this is the directional map, which shows us that in some areas, especially in the frontal or prefrontal cortex, especially in the right hemisphere, the enzymatic activity seems to be decreased, and in some other areas, especially the superior temporal gyrus and the somatosensory cortex, this activity seems to be increased. Right, so a very differential influence in this psychotic disorder context of the protein function. And this obviously then has differential effects also on the downstream pathways, right? So the areas with decreased activity or decreased expression of this protein should be downregulated, and the parts where it's upregulated or where it's increased should be upregulated. And that's, if we do this for all the clusters we got from the MCL clustering, right? I'm just going to go back for a second. So we did this then for these 7 clusters and summarize these maps. So all the protein inside the cluster that are functionally connected, aggregated the maps in a directional manner, and then come to this kind of, as I said, I just wrote this up today, so TV-based model of psychosis transition. And we have some brain areas that are implicated in this mechanism, especially the hippocampus, which is nothing new, which we saw earlier, which is especially implicated with the decreased plasticity and neurogenesis from the neural growth cluster. Then we have a differential effect of the dopaminergic pathway with increased dopaminergic signaling in the prefrontal cortex and parietal cortex, as well as the corpus striatum, but less dopaminergic signaling in the cingulate cortex. And kind of as a counterpart, with decreased glutamatergic signaling in the prefrontal cortex and increased in the retrospinal splenial cortex, which is kind of part of the cingulate cortex. So again, if you think back about the different mechanisms, we saw that there's this kind of disparity or this kind of anticorrelative behavior between glutamate and dopaminergic signaling. So this also would make sense in a grand unifying theory. And furthermore, we saw that we saw in the prefrontal cortex, that's where obviously most of the stuff is happening. So this is kind of the disease hub. We saw next to the dopaminergic increase in signaling, dopaminergic signaling, the decrease in glutamatergic signaling, also a decrease in GABAergic signaling, which was kind of the intermediary between these two neurotransmitter systems. And some decreased GABAergic signaling in the visual cortex. And which I cannot make sense of right now, a decreased inflammation in the dorsolateral prefrontal cortex. As I said, this is a very preliminary work in progress. But now we can have kind of this spatial pathway model of the mechanism that could explain the transition to psychosis, which is very nice in my view. And especially when we, so just to reiterate, the areas that are implicated in this model from Lieberman et al. Are also part of our model. And we actually extend this model with the parietal visual cortex and the cingulate cortex, which totally makes sense. And what especially makes sense is if you think about the symptomology of psychosis, right? So positive symptoms, increase in dopaminergic signaling in the prefrontal cortex, negative symptoms in this highly emotional cingulate cortex area with decreased dopaminergic signaling. Totally makes sense. Yeah, so if you think about this, how can we use this? So we have this spatial pathway for the transition to psychosis, and can this now actually feed into brain simulations, right? And Leon talked about this, and there's plenty of studies that show that regional heterogeneity actually improves and impacts brain dynamics in many ways, and especially it makes it biologically more sound, right? If we not just have a model of psychosis, but actually have a differential, heterogeneous, spatially differential model of the disease with different pathways impacted in different ways. There's a plenitude of how to model these heterogeneous alterations, for example, using the alterations in the dopaminergic pathway in a heterogeneous multiscale model, then actually modeling or just modeling specific parameters in TMP. And yeah, using local hyperexcitation parameters to model maybe the excitotoxicity that we see in areas with too much glutamate, or decrease in inhibitory parameters in regions where we decrease in inhibitory parameters in a region where we have less GABAergic signaling. And this is kind of, yeah, this was always our goal and our framework to use all of these knowledge mapping and knowledge sources to be able to infer simulations and infer knowledge directly from all of this without actually knowing anything, basically. So there's so much knowledge out there. There's so much databases, so much knowledge bases. We just have to use them properly. And if we see we have this empirical data agnostic to any hypothesis, we can use TU-based to decode it in a meaningful manner, getting us spatial pathway alterations that can then actually be used by the virtual brain, by TUB in simulations to test specific hypothesis, neurophysiological hypothesis about psychosis and about the transition to psychosis. So this part we still need to do, but I'm very confident that this will all come together nicely in the end. Okay, so these are my references. I just want to thank everyone in our lab, especially, of course, the ones I worked most closely here in this project. So Kiret, Leon, and Leon, but also Christoph, who helped us a lot in getting a better understanding of the interplay of the different neurotransmitter systems. And yeah, so that's it.